Okay, so now we're going to look at the role of blood pressure. So the kidneys play a very important role in blood pressure because they release something called renin. And renin's released by something we call the macula densa. And I'm just going to take you to a couple of pictures here first. Okay, so first of all, there's something here called the juxtaglomerular apparatus right here. And if you look at it here, you can see that there's a group of cells within that called the macula densa. So that's where renin's released, and it's right between the afferent and the efferent arteriole. And the only time renin's released is when we have a decreased GFR, glomerular filtration rate. In other words, our urine output's dropping. Okay, and because that's also because of your blood pressure probably dropping. See, the kidneys are on the B team. They think they're on the A team, but they're on the B team. So when we need our blood supply to be saved for the heart, the lungs, the brain, the liver, the kidneys get compromised. But that really makes the kidneys pretty mad. So what they do is they go through this whole series of enzymatic reactions. So they're only thinking really about themselves in that. So renin's released from these cells called the macula densa, and it promotes a number of enzymatic reactions that eventually produce a compound called angiotensin II. And it's a powerful vasoconstrictor. In other words, it makes all your blood vessels contract. Okay, constrict. And this also stimulates the release of aldosterone. And if you remember from the endocrine system, aldosterone increases our sodium reabsorption and therefore water, and it dumps potassium. So okay, so let's take a look again. So here's the distal tubule, here's the proximal. So you don't have the capillary around this right now. So here's the proximal tubule, here's the distal tubule. And this whole connection in here is called the juxtaglomerular apparatus. Okay, and it's nestled between the afferent and the efferent arteriole right there. And so there's the macula densa that we're talking about. And collectively, we call this whole business here the juxtaglomerular apparatus. Okay, so there are two negative feedback mechanisms that are involved in this regulation of glomerular filtration. And they're triggered when we see this decrease in filtration. And when the filtration rate decreases, so does the concentration of chloride ions. And that's just something else that happens. So in response, then, these cells, the macula densa cells, right here, these cells, signal the muscle of the afferent arteriole, this one, the afferent, the ingoing arteriole, to relax. Okay, and that dilates the vessel coming into the glomerulus or into the nephron. This increase in blood flow to the glomerulus increases the pressure and therefore the filtration rate. So it also causes this to dilate, so we get more blood flow in here and hopefully more filtrate. So meanwhile, the macula densa also signals the release of renin. And renin, remember, gives us this release of renin, which becomes angiotensin II, constricts all the smooth muscle cells in the efferent arteriole. So this one relaxes, and this one then also constricts. So we get blood backing up in here, hopefully to increase the filtration rate. So remember we talked a little bit earlier and we said, if we put all the water that goes through our kidneys in a day, we'd make about 45 gallons of filtrate, which is really impossible for us to lose all that. But our urine output actually is 1 to 1.5 liters a day approximately, and that varies with how much water we drink and how much fluids we have. So urine, when it's leaving the body, doesn't have the same as when it enters into the proximal tubule or the filtrate. Tubular reabsorption is the process that alters this content. I like to think of it like a reclaiming process. I'm just going to find a picture here. Okay. So remember that the efferent, or the outgoing arteriole, is narrower. Okay, and so most tubular reabsorption occurs here in the proximal tubule. And throughout the tubule at various points, we have different kinds of transportation, and we move molecules back into the blood along with water at various points. Taking a look here, here's an example. You can see the holes here in the filtrate and how it would occur. Okay, and so waters are reabsorbed because of osmosis, so we're sucking it back into the blood. While in the distal tubule, it's impermeable to water. And active amino acids are not found in the urine because we want to save all our protein, and it gets pumped back into the blood by active transport. Most water is absorbed passively in the proximal tubule, but as sodium is reabsorbed, then remember, where salt goes, water follows. So there's an obligatory water reabsorption with the sodium. So when our blood says I need to pull back sodium, then it gets pulled back, and so does water. So 70% of sodium reabsorption is in the proximal tubule and via the active transport pump too. So when sodium is positively charged, it is positively charged, as we know. It's reabsorbed and attracts a negative charged ion due to this electrical chemical attraction. So we always have a negative and a positive together. So the negative clings on to the sodium, and usually it's chloride, but it could be bicarb, and that just slips in with it. So this reabsorption of sodium and other ions increases the solutes of the blood in the capillary, and that increases the osmotic pressure. So the movement of sodium creates a strong osmotic gradient. And remember, osmosis is really, I talk more about it sucking than pulling, but it's water pulling, or think water sucking. And so water moves by osmosis back into the blood, because water is obliged to follow salt. And that's what we always say. Where salt goes, water follows. So this sodium linked water flow is called the obligatory water reabsorption.